Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Optimizing Access to High-Quality, Standardized and Compliant leukopaks for the Development and Commercialization of Allogenic Therapies. I'm Lauren Coyle, an editor at BioInsights, and joining me today is Mackenzie Becker, who will review the key considerations for ensuring access to and consistency of leukopak products for allogenic cell therapy starting materials. After the presentation, we'll have a live Q&A session. We invite our audience to pose their questions to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. Now, I'd like to introduce our presenter. Mackenzie Becker has over 10 years of experience in the biopharmaceutical industry. She currently works as the Manager of Product Development at NMDP Biotherapies, in which her and her team develop cutting-edge products that align with market demands, efficiently delivering solutions in a fast-paced, rapidly growing industry. So without further ado, I'll hand over to Mackenzie to kick us off with the presentation.
Welcome everyone to today's webinar on optimizing access to high quality, standardized, and compliant leukopaks for allogeneic therapies. My name is Mackenzie Becker, and I'm the Manager of Product Development at NMDP Biotherapies. With over 35 years of global leadership, NMDP has been at the forefront of advancing cell therapy worldwide. We've managed over 130,000 cell transplants and over 36,000 annual cell and blood shipments, demonstrating our logistics expertise in ensuring life-saving products reach their destination safely. NMDP manages over 7,000 cell therapies annually, with over 1,000 lives that have been directly impacted by NMDP biotherapies. Our success is supported by the strength of our registry. With more than 41 million donors, the world's most diverse registry ensures that patients have the best chance of finding a suitable match. Additionally, NMDP Biotherapies leads as the most experienced cellular starting material vendor in the commercial launches of cell therapies. Let's dive into our first topic, the consistency of leukopak products. Ensuring consistency is crucial for the success of clinical studies and the commercialization of allogeneic therapies. We'll explore key strategies for achieving and maintaining consistency in leukopak production, including donor reliability and reducing barriers to donation. NMDP Biotherapies contracts with 19 experienced apheresis collection centers throughout the United States. These apheresis centers are strategically located to align with donor location density. This, in effect, reduces barriers to donation and also enables geographic diversity to deliver products reliably and efficiently. All of our apheresis centers are accredited by FACT and/or AABB and routinely complete quality system audits. Our requirements for accreditation and adequate quality systems ensures a higher level of quality and consistency in the collection of our products. NMDP Biotherapies offers access to a large pool of volunteer donors from the NMDP registry. With over 7 million HLA-typed US-based donors, our registry ensures extensive HLA coverage for compatibility. In the past year alone, we've added 330,000 new registry members, with 35% of those individuals representing ethnically diverse backgrounds. But what we've learned is while the size of your donor pool is important, what truly defines the consistency of cell therapy starting material is having donors that are not only willing, but also compatible with your therapy requirements. One of the first steps of donor consistency is demonstrating donor reliability. NMDP Biotherapies donors are recognized as some of the most dependable in the industry, with a 90% collection completion rate once a donor has been qualified as a match for therapy requirements. The 10% non-completion rate includes collections that were requested to be canceled by a client, accounting for approximately half the total, which just indicates that our overall reliability of our donors is even higher. We also acknowledge the importance of leveraging donors with a history of successful donations and outcomes. That's why we continually assess our donor engagement processes to ensure an exceptional experience for our donors. When surveyed, 94% of NMDP Biotherapies donors say that they would go through the entire donation process again. As one of our donors stated, anything I can do to help someone else is 100% worth the time. Now that we've explored the importance of an established apheresis network and donor reliability, let's now turn our attention to our solution, the standardized GMP leukopak. This offering is designed based on a standard collection protocol, FDA requirements, and backed by proven logistics solutions to ensure high quality. We recognize that the quality of the product is not the only thing that matters. It's also about getting the product exactly when you need it. Therefore, we've established our processes to enable us to deliver a client's first product in as little as 6 weeks. Regarding regulatory compliance, our base product offering is compliant to the FDA requirements outlined in 21 CFR Part 1271 and 21 CFR Part 211. Each NMDP Biotherapies donor is evaluated under strict donor eligibility. This also includes maintaining all the necessary records of donor information, collection procedures, and testing results. We follow strict adherence to these guidelines to ensure that there are no setbacks in obtaining necessary approvals, as well as ensuring the safety and quality of our products. As for the donor requirements, our donors must be between the ages of 18 and 60 years old. A standard health history screening is conducted prior to collection, which consists of an interview of the donor about their medical history and relevant social behaviors. Potential donors are also screened via testing of infectious disease markers, or IDMs, at CLIA-certified laboratories. A physical examination is also performed to ensure the donor's suitability for donation. The physical exam helps identify any underlying health conditions that could pose risks to the donor's health during the donation process. And it helps identify any signs of infectious diseases or other medical conditions that could potentially compromise the donated material. At any step throughout this process, if a donor fails to meet the specified requirements, they will be unable to proceed to donation. Our apheresis center network follows a standard protocol for collecting mononuclear cells via apheresis. This protocol ensures consistency across collection sites, with specifications including up to 12 liters or 3 times the total blood volume, use of ACDA as an anticoagulant, and a minimum cell count of 10 billion total mononuclear cells, or TMNC. While these specifications represent our minimum requirements, our standard collection process consistently outperforms these limits, with typical cell counts ranging between 14 and 22 billion TMNC. As for product testing, our centers perform a complete blood count with differential reporting and total cell viability testing as part of our standard. We've incorporated these tests as they act as great indicators for cell health and quality. Our minimum requirement for viability is greater than or equal to 70%. However, we see an average of 98% viability on fresh products. As for the logistics, our base offering includes domestic product transport within the United States, maintaining temperatures at either ambient or cooled conditions. Temperature monitoring is performed throughout the transport to ensure the temperature is maintained. We also utilize methods of either shipping products or hand-carrying products by our experienced volunteer courier network. In instances in which our products are shipped, oversight of the transport is provided through GPS tracking. Our logistics processes have demonstrated the ability to maintain a stable temperature for our products. We performed a stability study of the product over 72 hours at 2 temperatures, 2 to 8 degrees Celsius and room temperature. The data presented here represents a subset of the findings from this study. What we observe is minimal fluctuation in leukopak viability and viable CD3 and CD4 cells over the duration of the study at both temperatures. Overall, we do see more consistency at the product in 2 to 8 degrees through 72 hours than at room temperature. Error bars have also been included to indicate the range between the minimum and maximum values seen at each time point. We understand that developers may have needs beyond the requirements and specifications we just reviewed. To address this, NMDP Biotherapies has a platform of add-ons that can easily be selected to enhance the standard leukopak. The enhancements available are for additional regulatory compliance regions outside of the US, additional donor requirements, product processing and testing, and enhancements to logistics and delivery. Starting off with our regulatory enhancement. By selecting and adhering to compliance regions outside of the US, developers can facilitate access to other international markets. We typically see that the most common regions outside of the US are the EMA, Health Canada, and TGA for Australia. While these regulatory bodies share common goals, there are key differences in regards to their requirements. This includes items such as varying donor exclusion criteria and eligibility. The guidelines for these compliance regions include a focus on donor health status, which may prompt health history questions in addition to those defined by the FDA. Additionally, there are variations in required donor testing, with required time points that may vary from FDA requirements. Having the flexibility to customize donor criteria to match a therapy is essential to curate a donor pool that maximizes manufacturing and therapy success. An advantage of our access to the NMDP registry is that we have the ability to maintain large donor pools with key donor demographics. Some of these demographics that may be customized are specific age range, birth sex, self-reported race and ethnicity, HLA type, blood type, BMI, and CMV status. Additionally, donor testing beyond what is required of the FDA or an international compliance region can easily be incorporated. NMDP Biotherapies has a catalog of available tests contracted at our qualified labs that can be selected, and we also have capabilities of drawing additional donor samples if clients have phenotype testing or other proprietary testing that they would like performed to optimize their manufacturing processes. Cell therapy developers may also be interested in additional quality control measures, which can be accomplished through product processing and testing add-ons. For fresh products, in addition to the CBC with differential and viability testing, sterility testing can be performed via BACT or BACTEC methods to detect any presence of microbial contamination. Additionally, we have standardized a flow cytometry panel for mononuclear cell products. This testing is particularly valuable for therapies reliant on identifying and quantifying specific cell subsets, such as cytotoxic or helper T cells and NK cells. The standardized leukopak can also be cryopreserved to provide more flexibility in scheduling manufacturing processes. We follow a standard cryopreservation protocol that was designed to automatically include sterility testing and a post-processing CBC with differential. However, additional testing options, such as the standard flow cytometry panel and mycoplasma and endotoxin testing, can be selected to further ensure the safety and efficacy of starting material. The final enhancement area is for customized logistics and delivery. One available solution is the ability to transport the collected product internationally. NMDP Biotherapies utilizes existing international relationships and regulatory expertise to facilitate these shipments, proactively identifying requirements such as any necessary accompanying documentation and labeling. Also, if a client requires expedited delivery faster than our standard service levels, we can assess and arrange a process that can accommodate this need to ensure on-time delivery for manufacturing production. Now that we've covered the key attributes and flexibility of the standardized leukopak, let's dive into one of its unique regulatory features. As I mentioned earlier, our team has the most extensive experience among cell therapy starting material vendors when it comes to FDA commercial approvals. This expertise not only sets us apart, but also provides insights into what additional offerings beyond the physical leukopak that could benefit cell therapy developers. A unique feature of NMDP Biotherapy's standard leukopak is the inclusion of a drug master file, or DMF, which sets it apart from other comparable products available on the market. For those who may be unfamiliar with a drug master file, it is a submission to the FDA that enables the DMF holder to provide confidential and detailed information about facilities, processes, or articles used in the manufacturing, processing, packaging, and storing of human drug products. I will note that a submission of a DMF is not required by law or FDA regulation. However, submitting a DMF when applying for FDA approval offers several benefits for both the applicant and the FDA. One of those benefits is that it can streamline the FDA approval process. Since the DMF contains detailed information about manufacturing, it can reduce the need for the FDA to request additional information. DMFs also provide assurance to the FDA that the manufacturing processes and facilities meet regulatory standards, ensuring the quality, safety, and efficacy of the product being submitted for approval. NMDP Biotherapies holds a Type 2 DMF, which is focused on drug substance intermediate, and material used in their preparation, or drug product. Specifically, our DMF is for mononuclear cells collected via apheresis and describes donor, cell collection, and processing activities that are performed for clients. This enables NMDP Biotherapies to maintain information and provide updates on our activities to the FDA directly to remove this burden from our clients. For clients partnering with us for their leukopak needs, we will provide a letter of authorization to the client to support their filing. At the time the letter is issued to the client, the FDA will also be notified. NMDP Biotherapies letter of authorization will identify the specific sections of chemistry, manufacturing, and controls that will be referenced that apply to the client's activities. Examples of the type of information provided includes the control of materials, specifications, and stability data. And before we move on to the Q&A, I want to thank everyone that has joined today. I've also included my contact information here, and if you have any further questions, please don't hesitate to reach out. Thanks again.
Thank you for that great presentation, Mackenzie. I'm just going to ask you to join us back on camera for the Q&A. Wonderful. So just to start us off today, the first question we have is, how do you choose your AC partners, and will all of them be used to collect for my protocol?
Yeah, this is a great question. When we evaluate and expand our network, there are two key areas evaluated for our apheresis centers. One of the areas is the geographic location of the AC. And as mentioned previously, we want to maintain centers that are near donor location density so that we can ease the donation process by minimizing travel for the donors when possible. Also, we want to make sure that we have enough coverage to ease our supply chain and to ensure that we can utilize centers that may be closer to manufacturing sites or the receiving location, and even to have multiple apheresis centers available if there's a winter storm or any other influence that would impact AC availability or delay any transport of the shipment. Another large consideration for choosing our AC partners is their capabilities. NMDP Biotherapies partners with centers that may have varying capabilities. They can meet all of our quality requirements and the collection protocol. However, they may not all be able to perform some of the cell processing attributes, such as cryopreservation, or even all the markers for our standard flow cytometry panel. So ultimately, any center in our network that meets the capability requirements can be utilized for a protocol. but we'll choose the centers based on availability and volume.
Amazing, thank you so much. So our next question here is, if I have add-ons to the standard, can I still reference the DMF?
Yes. So the drug master file is not just applicable to our standard or kind of base product requirements only. If there was any add-ons selected, like cryopreservation, NMDP Biotherapies will work with the client to include a letter of authorization that would cover the appropriate sections of the drug master file for the work that we are performing for the client.
Wonderful, thank you. And then what are your typical shipment turnaround times?
Yeah, so this will be dependent on the shipping configuration and the method, but we will be able to meet the following delivery times. So if a product is being transported domestically within the US, ambient or cooled products will be delivered within 24 hours of the collection, and cryopreserved products will be delivered within 48 hours for domestic products. However, if the product is being transported internationally, ambient or cooled products will be delivered within 48 hours, and cryopreserved products will be delivered within 72 hours of collection.
Amazing, thank you. And what are some of the big areas of considerations for starting material you want to use, if you want to use it internationally?
One of the first areas of consideration is the use of differing test kits for donor testing. So some of the infectious disease markers or IDMs required to be tested for the donor may be similar to the FDA, but there are regions that may require different test kits to meet each region. As one example, one of the EMA directives that NMDP Biotherapies follows specifies the use of CE marked test kits whenever applicable, which is not a requirement for FDA. Also, the timing of the donor testing for the IDMs may vary from what's defined in the FDA. As an example, one drop time point for the FDA requires testing within 7 days prior to collection or 7 days post-collection. And if you were also requiring Health Canada compliance, that required sample time point is 30 days prior to the collection. Therefore, if you were to collect the donor sample between that 30-day and 7-day time point prior to collection, or even after the collection occurred, it would only meet one of these regions' requirements. And the last sorry about that. One more consideration for these testing laboratories is that there may be differing accreditations that the lab must follow if they were doing it outside the FDA compliance region.
Wonderful, thank you. And can you elaborate on your regulatory support for regions outside of the United States?
Yeah, so I've mentioned previously we've built in solutions for Health Canada, EMA, and TGA in our standardized leukopak, but I will also mention that we have additional experience outside of these regions. We've just built these in because these are the most common ones that we've seen. One of these regions that we've have experience with is Japan's Ministry of Health, Labor and Welfare, or MHLW, but ultimately, for any compliance region outside of these standard international compliance regions, NMDP Biotherapies can work with you and assess your needs and build a custom solution.
Wonderful, thank you. Can a donor center only be AABB accredited, or does it need both FACT and AABB accreditation?
Yeah, so this accreditation is actually applicable to our apheresis centers in our network. We do not require both accreditations, but we do require the center to be accredited to at least one.
Wonderful, thank you. And I think we have time for one more. So for process development, it is essential that the donor gives good expansion and good transduction. Is there any pre-screening that can be done or a parameter that can be screened to ensure good quality expansion?
Yeah, so that's where we have more of our features for customization for our donors. If there's any donor characterization, I will also plug that we have some great bioinformatics available through NMDP Biotherapies that we can actually do some proactive work into your donor pool, into population genetics to make sure that we are modeling and designing a cell bank that meets for you. And we can also discuss any client sample testing. Maybe there's a really good indicator that you have that's a pre-screening or parameter for the donor testing, which we can implement as long as we arrange the transport to your qualified lab or your location.
Wonderful. Thank you, Mackenzie, for answering those questions. But that's all we have time for today. So any questions we didn't get to, we will reply to by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. All that's left is to thank you, Mackenzie, once again for a great presentation, and thank you to our audience for listening today, and we hope you'll join us again soon.